This is a production of Cornell University.
Thanks for coming. Can you hear me? Can the people in the back hear me okay? Yes. Great. So as Eric mentioned, today I'll be talking about my dissertation research and focused on this topic of the mysteries of the marsh, looking at soil-borne plant pathogens in marsh wetlands and their impact on invasive and native plant species. And through my dissertation research, I've been really trying to answer the question of whether soil pathogens are contributing to the success of invasive plants in marsh wetlands. And a lot of the work that I've done has just been laying the groundwork to answer this question. So today's seminar, I'm going to be focusing on presenting to you a few experiments that are answering questions that I think really enable us to start to answer that question. And that is, do invasive plants change soil in a way that decreases seedling survival? And if so, how? Which oomycetes and fungi are present in marsh wetland soils? And are these oomycetes and fungi pathogens of marsh plants? And if so, how virulent are they? So in this picture, you can see two fields right near where I work at the north end of Cayuga Lake in the Montezuma Wetlands Complex. Here on the left, you have something that perhaps is a little more familiar to those of us in this room and in this department. This is a wheat field. And on the right, this is a marsh wetland. So you can see a lot of differences right off the bat between these two areas. But I think the biggest difference, and kind of relatedly the biggest difference between agricultural areas, which are highly managed, and less managed, more natural areas, is diversity. So this diversity is both the richness and evenness of species. So the richness being the number of species that are there, their evenness being their abundance relative to each other. And it also happens on both a genotypic and a spatial scale. So if you think of, in this cornfield, there's probably just one genotype there, one species, but also one genotype. Here you're going to have a lot of different species, a lot of different genotypes within each species, and the spatial heterogeneity is much larger. So one exception to the rule that natural areas tend to be really highly diverse are invasive plants. And by invasive plants, I mean that these are non-native plant species, so they've evolved somewhere else. They've come to a new place. They're rapidly expanding their range. And for the most part, they have some negative consequence, either on humans economically or on some ecosystem services, like animal habitat or biodiversity of plant species. So in this picture, you can see two invasive plants dominating this marsh wetland. In the foreground here is Phalaris arundinacea. It's reed canary grass. In the background is Phragmites australis, Phragmites or common reed. And you can see that they're both growing in really dense stands, excluding the growth of other plant species. So if most natural areas are highly diverse, how are invasive plant species able to do this? Able to grow so densely? And a lot of ecological research has looked into plant traits of these plants, so the fact that they're able to grow really fast, they can grow taller, shade out other plant species. But increasingly, people are looking at the effect that they're having on the soil. Many invasive plants change soil properties, abiotic and biotic, in ways that put them at an advantage and other competitors at a disadvantage. This is called soil conditioning. And I'm interested in how that's happening with invasive plants in marsh wetlands, and is that different from what native plants are doing? So just a little background on what's happening in most kind of native plants, natural systems. You have density-dependent recruitment of seedlings, which contributes to the high diversity that you see. So if you imagine the probability that a seedling will survive and the density of that particular plant species in the soil, there's going to be higher survival when that plant species is at low density and lower survival when that plant species is at high density. And the way that people found this out was in the 1970s. Two ecologists, Jansen and Connell, saw that in tropical forests, if there's a parent tree species it's seedlings. There's going to be a lot of species near the parent, and that's kind of what I'm considering a high density of that particular plant species. And then there are going to be a few seedlings that are further away at low density of that plant species. And that the seedlings near the parent tree die or do very poorly, and those that are further away at a low density have a much higher probability of survival and eventually fitness. So when they first found this, they thought that it was probably mammals, some kind of seed predators, but increasingly people have found that no, it's plant pathogens in the soil that are causing these effects. So related to that, I wanted to know what's going on with invasive plants if they can avoid those density-dependent effects in the soil. So in this picture, I really like this because this is Phragmites, beautiful tall grass, and right in the background here you have another beautiful tall grass, corn. And so a lot of the fields that I work in are right next to agricultural fields. Both of these are monocultures of very dominant, very tall grass species. And so those of you who work in agriculture know it's really hard to get corn to grow like this. It doesn't naturally want to not have weeds. If you didn't manage it, if you didn't rotate crops, you'd have big problems both with weeds and diseases. So how are invasive plant species, and this plant species in particular, how are they able to do this with no inputs? And what people are finding is plants have different pathogen interactions in their native and invaded ranges. So if you think back to those density-dependent soil effects and the role of soil pathogens, that they are playing a role in the success of many invasive plant species. So one example of this, and why we think this is happening, is that if you grow the same plant species, for the most part in the same soil year after year, it's going to do more poorly each time. Those of you in agriculture know that this is the case. You have to rotate your crops. The same happens with native plants. So if you look at plant soil feedback, and over time it'll be lower and lower. Plant soil feedback being the term for how much the soil kind of feeds back to those plants' survival and success. So native plants do worse. Invasive plants don't. And so people have found that they are neutral when grown in the same soil, or maybe even benefit from it. And so they're having some kind of different interactions there for native plants. And what we think is happening, one hypothesis is called enemy release, and that is that when invasive plants come, they leave behind pathogens from their native range, and it takes time for them to accumulate and acquire them when they invade. So if you imagine this invasive plant in its native range, it's going to have a diverse group of pathogens, including some soil pathogens that I've illustrated here. When it leaves those behind in an invaded range, it can sometimes do better because it's not experiencing those density-dependent negative feedbacks. So one classic example of this was with Prunus serotina. Our black cherry is invasive in Europe, and it's able to grow in really dense stands, unlike here. And the reason for that is that it's encountering less virulent soil pathogens, particularly of the genus Pythium, that don't have the same density-dependent effects on seedlings. Those seedlings are able to survive to adulthood, and that's why it can grow in such dense stands. So if that were happening in a marsh wetland, you'd expect to see invasive plant species less susceptible to pathogens in the invaded range than native plants. And you'd expect that native plants would accumulate more virulent pathogens than invasives. Another hypothesis for how invasive plants are interacting with pathogens in a way that puts them at an advantage is that they're somehow changing the pathogen population when they invade in a way that puts native plants at a disadvantage. So this could be them bringing in pathogens with them that would then spread into a native community, or it could be just something about their arrival changes the pathogen community in such a way that native plants have higher disease and decreased survival. So here we've got a plant coming into a mixed plant community, an invasive plant, and it's bringing with it some pathogens that would then spread into the native plant community. And this hypothesis is called spillback or spillover, and a few examples are with invasive grasses in the western United States. Several are known to bring with them viruses that will then spread into a native plant population and increase the disease that they're experiencing. Another example that doesn't involve a plant bringing in anything with it, but just changing the existing pathogen community, is with cheatgrass, also in the western United States. So cheatgrass is a prolific seed producer. After its arrival, there's a lot more seeds in the soil. The abundance of those seed pathogens really increases, and the native plants have a hard time outcompeting cheatgrass. So if that were happening, you'd expect to see a pathogen community changing after an invasive plant colonizes, and you'd expect maybe increased abundance or virulence of pathogens after invasion. So here are a few different hypotheses for how pathogens could be driving the success of invasive plants, both because invasives escape pathogens from their native range, it takes time for them to accumulate them in an invaded range, and they might be able to change the pathogen community when they invade. So I want to know how important these are for invasive plant species. And particularly, I want to know what's going on in marsh wetlands. So most of the studies I just described take place in forest and grassland systems. But marshes are really important for understanding invasive success for a number of reasons. First off, they're highly invaded systems. So if you think about the kind of most important invasive plants in North America, Phragmites, Australis, reed canary grass, purple loosestrife, a lot of those are in marsh wetlands. So understanding what's going on there, I think, would be key. Second, marsh wetlands provide important ecosystem services, water quality, habitat. In this area, a lot of the migratory birds pass through where I work at the north end of Cayuga Lake, so they need that habitat. There's also the issue of the kind of different plants that are there. These invasive plants are preventing them from growing, so the biodiversity. Also, there are lots of oomycetes and fungi in marsh soils when people have looked. So in Europe, they also have marsh wetlands. In fact, some of our invasives are native there. And so when people have looked, they've found really abundant communities. So I want to know if that's happening here. Seed bank establishment and seedling establishment are really key for the success of a lot of these plant species. So paired with the fact that there are a lot of soil oomycetes and fungi, I think it's probable that there's some interaction going on where pathogens could be determining what the plant communities look like. And then finally, some of the pathogens that are known to affect the dieback or kind of death of a lot of the reed species in Europe are oomycetes, similar to what I think we find here. So I want to know if they're also playing an important role in our marsh wetlands. So before I begin, I just want to introduce you to a few of the plants I'll be talking about. First, Phragmites australis. I've mentioned it before. It's non-native, it's European, and I think it came here in the late 1800s, and since it's been replacing a lot of native plant species. You've probably seen it on the roadsides. It's kind of lining a lot of the highways, but it's also important in wetlands. And it forms these large clonal patches, like you can see here, extremely dense and quite tall. It can get up to 13 feet. And one of the interesting things about Phragmites is that there are both native and non-native genotypes here in North America. So that's a good species for us to be looking at because it gives us a closely related pair of native and invasive. I'll also be talking about a couple other invasives, Phalaris or Indonesia, reed canary grass, and Lithorum salicaria, purple loosestrife. You've also probably seen these around. I think this one is particularly beautiful, even though it isn't invasive. And I'll be mentioning a few native plant species. Epilobium glandulosum is a herbaceous native plant, Calamagrostis canadensis, a grass species. In the experiments I'll be talking about, I'll be using just a wide variety of different native plant species as kind of a proxy for what's going on in that community, with less attention to what particular hosts or plants are experiencing. So the first question, I think, for determining if plant pathogens and soil pathogens in particular are playing a role in the success of invasive plant species is seeing, does the invasive plant change the soil in a way that decreases seedling survival? So I want to note Phragmites australis, that invasive plant I mentioned initially, is changing the soil. And this could be abiotic or biotic. Some invasive plants exude allelopathic chemicals that are toxic to other plant species. But I'm particularly interested in the pathogens, so I want to know, is it reducing seedling survival of other plants? And is this caused by soil, fungi, and oomycetes? And so to get at this question, there are a couple different experiments I'll mention right now. First, I planted seedlings of 8 native plant species at 4 sites. So here you can see me carefully measuring some seedlings. At each site, I cleared plots within and outside of the stand of Phragmites. So this, you can see the dense Phragmites around me. And the reason I cleared those sites is that I wanted to exclude factors like shading and crowding that people typically think are why Phragmites is so successful, and just look in the field at the soil. So after a month, I assessed seedling survival. And here's a plot outside of the Phragmites. So you can see I've drawn a square here just so you can see where the plot is, and the kind of much more diverse vegetation outside of the Phragmites relative to inside of it. And you can see some little patches of green here, and what those are the seedlings that I transplanted. This is one of them, a grass. And what happened was that there was overall lower survival in Phragmites stands. So this is the proportion of seedlings surviving. This is plant species transplanted, so a number of different plant species here. The yellow is inside and the green is outside. So overall, there's a clear trend that things did better outside of the stands than inside. There were some site-specific effects, so I think these soil changes take time to accumulate. So one of my sites was a more recent invasion, and it didn't have those differences. And because of that, it's only significant for a couple plant species. But overall, there's a clear trend that Phragmites seems to be doing something to the soil that decreases the survival of other plants. But what is that? So to answer what exactly is happening in the soil, I did a secondary study with my collaborator Laura Martin and some members of Aaron Blossey's lab, looking at seedling survival in a more controlled container setting. So I took soil, or we took soil from these Phragmites common gardens that had been conditioned, grown with Phragmites, and then planted 10 different marsh wetland plant species in the soil. And half of these containers were treated with a broad spectrum chlorothalonil fungicide that should be somewhat effective against both fungi and oomycetes. To see if you exclude those from the soil, do you still see Phragmites having a negative effect on the survival of these plant species? And so as you can see, there were many containers. And what we found is similar to in the previous study. For some species, but not all, Phragmites did have a negative effect on seedling survival. So the brown is potting mix, the yellow is Phragmites conditioned soil, and the green is that soil with fungicide added. So the asterisks represent a significant interaction or contribution of that factor to the proportion of seedlings surviving. So you can see that for a few of these species, there was a significant decrease when they were grown in the Phragmites soil, but for pretty much all of them, the fungicide increased survival. And in many cases, it brought survival kind of back up to the level that it would be in potting mix. So if you see right here and here, the fungicide is able to take away those soil effects. So to answer that question, does Phragmites soil conditioning, or the changes that it makes to the soil, affect seedling survival, and how? We did find that it decreases survival of some plant species, but not all, and that this was caused by soil and fungi, or soil oomycetes and fungi for some plant species. So we know that these fungi and oomycetes are playing a role, but we don't know which species these are. So my next question was trying to identify which oomycetes and fungi are present. And my hypotheses were that they'd be diverse and abundant, both for fungi and oomycetes, just based on what people have found in other systems. But that there would be different pathogens, or actually not pathogens, fungi and oomycetes, associated with different plant species. The pathogen part will come later. So for the oomycetes, we sampled field soil at 4 sites, both Phragmites invaded and non-invaded. And we isolated oomycetes using a leaf bait method. So here you can see there's some soil at the bottom of this tube. It's filled with water, and then there are leaves at the top, and that selects for oomycetes because it requires swimming zoospores to reach and infect the leaves. And then we used warp-selective media. And after isolating, we extracted DNA from the isolates and sequenced the ITS 1 and 2 region of the rRNA operon. And so we got about 500 isolates, and as you can see, they're really dominated by Pythium species. So some Phytopythium, some Saprolegnia, but mostly Pythium. And if you divide this up by which Pythium species were we seeing, Pythium inflatum, Pythium dissimile, Pythium heterothalcum, and Atranthoridium, and Aquatile, Angustatum, those are kind of the dominant species. But it's worth noting that a really large percentage, about 20%, were species that only had less than 10 isolates. So not only are we seeing a lot, so we're seeing a lot of different species, we're seeing some dominant species, but a lot of minor ones that still might play a role. So if you compare how frequently these species were isolated from Phragmites invaded and non-invaded soils, you do see some differences. Some species, this is the difference in isolation frequency right here. So a 0 would mean that there is no difference in how frequently they were isolated from the 2 soils. These are more common in non-invaded, and these are more common in the Phragmites invaded soils. So you can see that some species, Pythium irregulare, Pythium phragmitis, are more common in the non-invaded soils, while others, like Pythium torulosum and Pythium iranomnes, were more common in the invaded soils. So to look at fungi, we took a little different approach. You can see my fantastic field assistant here planting containers in January. And we buried seed baits at 10 sites using seeds of 9 different plant species. And these seeds overwintered in the field, and in the spring we collected them, extracted the seeds from each container, and then we isolated fungi. So you can kind of see this is a seed from one of those containers, and there's some hyphae growing out. So took the hyphal tip from each isolate growing, and repeatedly transferred it so we could make sure we had a pure culture, and then we identified these isolates, sequencing them the same way. And so far, I have a larger culture collection of around 1,000, but so far we've sequenced about 266 isolates, and we're seeing a lot of different fungal species. So here's Alternaria, Phoma, Epicoccum, and Fusarium seem to be the dominant genera. But there are a lot of others, especially in this group, genera with 1 isolate. And so if we look at this by plant species, so remember we had those 9 different plant species that we're isolating these fungi from, you can see different fungi are associated with the seeds of different plant species. So each of these circles is roughly the number of isolates that we have from each of those plant species. So this is Phalaris and Arundinacea, the invasive that I mentioned, the canary grass. So that has a pretty even mix of different genuses of fungi. But for Phragmites, it's very dominated by Phoma, also some Alternaria, and Carixpanchia, which is a native sedge. The dominant genus is Alternaria that we're finding associated with these seeds. And again, I did surface to sterilize the seeds before putting them into these field sites, but it's possible that some of these fungi are coming in with the seeds rather than being acquired from the soil. But still some clear differences. So which fungi and oomycetes are present in marsh wetland soils. We did indeed find diverse and abundant oomycetes and fungi. So for oomycetes, the dominant genus was Pythium. For fungi, the dominant genera were Alternaria, Phoma, and Epicoccum. And there did seem to be different species associating with different plant species. So some fungi and oomycetes were more common with particular plant species. So next, I want to know, sure, we know what's there, but what are they actually doing? So are these pathogens of marsh plants? And if so, how virulent are they? So my hypotheses going into this, just based on the types of genera that we were seeing, Pythium in particular, is that we find a lot of generalist pathogens. But thinking back to some of those hypotheses for why invasive plants are so successful, I expected that native plants would be more susceptible and that isolates from the native species would be more virulent. So to test this, we set up bioassays for both fungi and oomycetes, and here's the structure of that. I added or grew isolates of each on an agar plate and waited until the mycelia had grown out far enough, and then added seeds of different plant species to the surface. So this is a pretty artificial assay. It's definitely not what's going on in the field, but it at least gives us some idea of whether or not, if given all of the opportunities, these isolates can be pathogenic. So by pathogenic, I mean that they decrease seedling survival when inoculated relative to a control that's uninoculated. And then for virulence, I'm defining that as the degree of that decrease. So in this top frame, you can see a control uninoculated plate growing with seeds of Phragmites australis. In this one, you can see a non-pathogenic isolate, so all of those seedlings are doing great. Here is a low virulence isolate, so one that does decrease the survival of these seedlings relative to a control, but that there's still pretty high seedling survival. And then in this bottom frame are some high virulence isolates, so few, if any, of those seedlings are still surviving. So we measured seedling survival at the end of 3 weeks, and this is for the oomycetes. I'll show them first, and then the fungi second. So think of this as a heat map with the different Pythium species here in order of their relative abundance, so how frequently we isolated them from the soil. And here are the plant species that we tested. So these ones in green are native, these ones in yellow are invasive. And what you can see is that some plant species are highly susceptible. So the darker the red, the kind of bigger impact, the more virulent those pathogens are. And those with a significant decrease from the control, so those I deem pathogenic, are shaded in any shade of red. So those that are white did not appear to significantly decrease seedling survival. So the really dark red, you can see here, Epilobium glandulosum, one of the native plant species, was the most susceptible to these pathogens. And I think this invasive Lithium salicaria was kind of also extremely susceptible. And you can see when inoculated, it had very low seedling survival. The least was Phalaris omninecia, an invasive plant species. You can see none of the isolates we looked at significantly decreased seedling survival. Some of these isolates species were highly virulent. So Pythium velutum, Pythium phragmitis, you can see that pretty much across the board they decreased seedling survival to around 0 for the plants that we tested. Whereas others were occasional pathogens or maybe very weakly virulent pathogens, such as Pythium citrinum, right here, as well as Pythium aquatile. So another way to look at that same data would be spreading it out, just looking at 2 plant species. This is a native plant, Epilobium glandulosum, the one that I mentioned is really susceptible to these pathogens. And this is the invasive Phragmites in yellow. And proportion seedling survival, Pythium species tested, with asterisks for the significant, those that significantly decrease seedling survival relative to control. So what you see are a lot of asterisks. So a lot of these interactions are pathogenic. those species do significantly decrease survival, but the range of that is really broad. So some of these are just acting as a pathogen. So this is Pythium angustatum. You can see that the survival for Epilobium is pretty much 0 when inoculated, whereas for Phragmites it's very high. So those are having that Pythium species is having a really different effect on those two, whereas others like Pythium dissimile is very similar. But some, although it's a pathogen for both, it's a far more virulent pathogen for one rather than the other. So if you look at Pythium accumulatum here, you can see that Epilobium glandulosum, when inoculated, has pretty much 0 seedling survival, whereas Phragmites, despite the fact that it's significantly reduced relative to control, it's not that virulent of a pathogen. So if we divide this up by where these plant species are coming from and look at the effect of all of the Pythium species, green being native and yellow being invasive, you can see that the proportion seedling survival, it isn't clearly higher for the invasive plant species. So if you think back to Emily release, it would say that these invasive plants are going to be less susceptible to pathogens. And we're not seeing that across the board. One of the most susceptible plant species, Lithium salicaria, is invasive, but at the same time, the most resistant, Phalaris onanissia, is invasive too. So they're not clear-cut divisions between native and invasive species. We can take this one step further and look at individual isolates of a given species that are taken from either the Phragmites invaded or non-invaded soils to see if something about Phragmites being there makes this more virulent community. And so we did this for, I think, 8 different species. I'm just showing one here, Pythium and Trantheridium, but took isolates from both the Phragmites invaded and non-invaded range and tested their pathogenicity and virulence to a number of plant species, which are in different colors here. And the green are natives, and the yellow is the invasive Phragmites. So what you can see is that there's not any big difference between the isolates from the Phragmites invaded and the non-invaded. They respond really similarly for all the plant species we looked at. So we're not seeing Phragmites cultivate an extremely virulent pathogen community, at least within species. So for the fungi, here's another heat map with the isolate source plant, the plant that were the seeds that we're getting these fungi from, and then the plant species we tested along here. And you can again see that Epilobium glandulosum, that herbaceous native species, is the most susceptible, with the darkest and most red here, the biggest decrease in seedling survival. And the least is Muhlenbergia glomerata, which is a native grass. These two invasives here, Phragmites and Lithium salicaria, are kind of in the middle. And isolates from some plant species might be slightly more virulent than others, but it's really a mixed bag. So if we again divide this up in the same way as the previous group with oomycetes and look at those individual fungal species by these two plant species, Epilobium glandulosum and Phragmites, we see similar things, that a lot of these significantly reduce survival relative to the control, but there's a big difference in the virulence, so the degree to which that negative effect is happening. The most virulent pathogens are these Fusarium species, no surprises there for those of us in the plant pathology department, as well as some of these alternarias. But it's not necessarily kind of uniform across species, so this alternaria species, it significantly reduces seedling survival for both Epilobium glandulosum and Phragmites, but it's having a bigger effect on Epilobium. So we do the same thing and look at the kind of sum of all of those fungal species by plant species and by plant species origin, with the native species in green and the invasive in yellow. You can see that again, you're not seeing clear-cut differences between the native and invasive species. Some of these invasives are very susceptible and others are not. And in this case, the most susceptible is still Lithium salicaria, and the least is Phragmites australis. So, Phragmites. So getting back to our initial question, are these oomycetes and fungi pathogens of marsh plants? And if so, how virulent are they? We did find a lot of generalist pathogens, so they decreased seedling survival of a range of plant species, but there were differences in their virulence. And when looking at whether native plant species were more susceptible than invasives, this did seem to be true for some invasive plant species, but not for all. So Phalaris or Anaceae was pretty resistant to those fungi and oomycetes, but Lythrum salicaria, another invasive, was highly susceptible. And similarly, the pathogens from the native species weren't more virulent than those from the invasive. So, to summarize, we found that Phragmites changes the soil in some way that decreases seedling survival. And when we looked at that further, we found that that was because of oomycetes and fungi in Phragmites conditioned soil that were decreasing seedling survival. We found diverse and abundant oomycetes and fungi in these marsh wetland soils. And the dominant genera of these were Pythium, Alternaria, Phoma, and Epicalcum. And we found different pathogens associating with different plant species. So how did these fit into those hypotheses I mentioned initially about how invasive plant species might be interacting with pathogens? So if we think back to enemy release, where part of the reason why an invasive plant would be successful is because it's leaving behind pathogens in its native range, and it's not acquiring them in its invaded range, we would have expected to have seen that invasive species were less susceptible to pathogens in the invaded range than native plants, and that the native plants were accumulating more virulent pathogens than the invasive plant species. So did we see this? We did for some invasive plant species, but not all. So it's not a clear rule for these marsh wetlands that invasive plants are acting one way and native species are acting another way. So it does seem to be the case that Phalaris or Anaceae is less susceptible to these pathogens, but for Phragmites and Lythrum salicaria, it doesn't seem to be. So this also doesn't get into the question of, we're just looking in its invaded range. So this doesn't tell us what's happening in the native ranges of these invasive species and what those pathogen communities look like, but at least it gives us a little window onto what's happening. And then thinking back to the other hypothesis I mentioned, that something about the addition of this new plant species, this invasive plant, is changing the pathogen community in such a way that it would decrease the success of native plants, spill over or spill back. With that, we would have expected that the pathogen community changes after invasives colonize, and that it increases the abundance or virulence of pathogens after invasion. So did we see that? We did see that the pathogens seem to be different. If we looked at the oomycete and fungal communities of different plant species, they were different, but we don't know if they're necessarily more abundant or more virulent or what's happening with that and what effects those would have on the plant community. So I think that this research lays the groundwork for more work into what's actually happening in marsh wetlands. We now know which oomycetes and fungi are there. We have some idea of their host ranges and their virulence. But I think one of the things that definitely comes out is that marsh wetlands are a little different from the forest and grassland systems, where other people have looked at pathogen invasive plant interactions. And I think that there might be a few reasons for that. So when people looked at invasive plants in grassland and marsh systems, they tend to find a really clear divide between the native plants, which behave one way, and the invasives that are different. And here we're finding that's not the case, that each plant species kind of does its own thing. And I think that could be because marsh wetlands are just different from those other two systems. Marshes are, even the native species tend to be quick colonizers. They tend to grow in clonal stands, and they're just different from those species in forest and grassland systems. In addition, I think that we found a lot of generalist pathogens in these soils, which might be making it so that there's less host-specific interactions, pathogen interactions in marsh wetlands. But more likely, I think that if people looked at a broader set of invasive species in other systems, forest, grassland systems, they'd probably find the same thing, that different invasive plants are benefiting from different interactions. So some may be doing so well because they're growing really fast, others may be interacting with soil pathogen communities, and that you can't really make a blanket statement about how invasives are acting and how natives are acting. So with that, I want to thank everyone who made this research possible. Eric, thank you so much for being such a fantastic advisor. I really have grown through your mentorship. And this is where you have to sit with me because this is an exit seminar, just not a normal seminar, so I get to thank everyone. Mary Ann, right there, I don't know what I'd do without you in the lab. You're one of the few people who's as good in the lab as in the field. And Mary Ann really was essential for a lot of the, particularly the sequencing molecular work behind this research. Justin Lansafane, my undergraduate assistant right there, who is co-writing one of the papers I'll be coming from my dissertation with me and has really been a great help. My committee, Bernd Blasi and Allison Power, all of my many undergraduate assistants, everyone from the Blasi lab, Vicki, Nuzo, and Andrea, and then my funding from the Atkinson Center and the New York Sea Grant. And I also want to give a thanks to all of my colleagues, friends, family for your support. And I know that's everything from helping me in the field to listening to my practice talk. And here are my parents there, my sister, and they can tell you that I have been very into marsh wetlands ever since I was a little kid. So here is me doing approximately what I'm still doing at the age of maybe 5 or 6. So not much has changed there, but I do feel really grateful to have spent the past 5.5 years surrounded by such a great group of kind of plant pathologists, but also friends. So with that, I'll take questions, and thank you all very much.
This has been a production of Cornell University, on the web at cornell.edu.